Okay, we're going to continue our discussion now. There are a few points that I need to finish up with the Basidiomycota, or club fungus, the ones that form sexual spores that are on club-like basidia. Remember, that's the distinguishing characteristic of this phylum. One of the interesting things that these club fungus do is they grow in rings. Sometimes you'll go out in the woods, and there's a picture of this in your book that you should take a look at. And it is an almost perfect circle of these fungus growing. And inside the circle, there aren't any fungus. And in the immediate area outside the circle, there aren't any fungus. And this is what's known as a fairy ring. A fairy ring is an almost perfect circle of growing fungi, or growing mushrooms, inside which and in the immediate outside which there are no other mushrooms growing. And there's a lot of thoughts about why this occurs, but it's an interesting phenomena that you can take advantage of when you're looking for mushrooms. Basidiomycota also are responsible for some very interesting diseases. And one of the more interesting problems is something called rust. And this is a picture of the rust, and I wanted to show this, and I'm going to go through this in a minute because in your book they talk about 3 or 4 different types of spores that occur, and I wanted to point that out on this diagram. But before I do, I also want to talk about some other Basidiomycota genus and species. There's a group of Basidiomycota that are called puffballs, and the puffballs literally grow in round ball-like structures. Inside those ball-like structures is the spores, and then when an animal comes by and steps on one of these puffballs or bumps up against it, or there's a strong wind or some other trauma, the ball literally explodes or detonates, so to speak, and out puffs the spores. And this is the way that the puffball spreads its progeny in a greater and greater distance. Now, as I said, one of the more interesting species of the phylum Basidiomycota is called a rust. And rusts are parasitic. And the difficult problem with the parasitic fungus is the fact that they attack commercial grains such as wheat. This life cycle that I've shown you here is actually a wheat rust. And the other thing that's very interesting is they generally have 2 hosts instead of 1. Most parasites only feed on one host. But the parasitic fungi, particularly the parasitic rusts, can have 2 hosts, one of which is called the main host and the other is called the alternative host. Now, when it initially affects a plant, you start out with these red spores. And the red spores are called eurydeospores. And they are the first aspect of the infection that can occur. And as this goes along, then other spores form until you get around to this achetial spore. And these are yellow in color and can be spotted because of the yellow color. And they form what's called a cap or cup. And the spores are located inside here. And this continues in this infection in this way. Now, wheat rust can be quite a devastating disease for the wheat in the United States, and it's frequently treated to prevent this from happening. Another type of parasitic fungus is called a smut. And a smut, I always get tickled with these various names because smut is also a term that means bad literature. So a smut is a parasitic fungus that also infects many different kinds of crops, including wheat, corn, barley, oats, various other types of crops. Now, the second phylum is called Ascomycota. Ascomycota is distinguished from Basidiomycota in the fact that the sexual spores come from sacs as opposed to clubs. And the sacs are called asci, thus the name Ascomycota. Frequently, these groups of fungi are called sac fungi because of this sac formation. Now, the spores that are found in the sac are called ascospores. And yeast is one of the members of this group, and it is the single-cell organism that is the member of this group. Now, yeast is unique in the fact that it can reproduce asexually, and I'm not sure what that word is. It's a typo, sorry about that. But they can reproduce asexually in a form called budding. And essentially what occurs is you have a reproduction of the nucleus here. So you have two daughter nuclei, and at the same time that the nuclei is reproducing or dividing or undergoing mitotic division, and we will talk about mitosis at some point, there's a bud that forms at the same time. And then during this time, one daughter nucleus goes into the bud along with part of the vacuole, and the other nucleus remains with the parent. And then ultimately this separates and forms a scar right here, what's called a budding scar. And you have what's a daughter yeast and the parent yeast. Excuse me. And so we end up with two yeast that are genetically identical. And as I said, at one point on the yeast, there's evidence that a bud has occurred, and that's known as a budding scar. Now, we've talked about the metabolism of yeast before. Ascomycota are the group that undergo fermentation. And in the fermentation of Ascomycota, ethanol is the primary product. Remember, fermentation is anaerobic breakdown of sugar. And remember, there were two products that could form, ethanol and lactic acid. And in mammals and other normally aerobic organisms, lactic acid is what's formed. But in yeast, where fermentation is normal, the anaerobic breakdown results in ethanol. And so what you have here is sugar from some source enters the yeast, and the yeast produces three things: energy, about 2 ATP per molecule of sugar, ethanol, and carbon dioxide. The carbon dioxide is what causes the bread to rise. The ethanol is what puts ethanol in beer, wine, and various other drinks that use fermentation in order to form their alcohol. And then the energy is how the yeast get by. Now, what we do is, if we want more ethanol or a greater production of ethanol than what we get, is we take, say, a gallon of naturally produced ethanol, which has maybe 5 or 10% ethanol and the rest is water, and then we do what's called distillation. And it's actually called fractional distillation. And the reason it's called fractional distillation is because you use heat, and the fact that the boiling point of ethanol and the boiling point of water is different. Ethanol boils at about 80 degrees centigrade, and water boils at 100 degrees centigrade. So if you heat a mixture to 90 degrees, then ultimately all of the ethanol will boil off, and you are left with pure water. And what you do is you have a cooling tower above the boiling substance, and as the steam rises into the cooling tower, it condenses and it's captured, and then you have alcohol separated from the water. And this process is known as distillation. Now, it's called fractional distillation because you use heat fractions. And a lot of times when you use the term fractional distillation, what you're actually talking about is the removal of 3 or 4 different kinds of substances that all have different boiling points, then you're removing each fraction from the other. The third phylum is called Zygomycota. Zygomycota is the bread mold that grows on bread. And the distinguishing characteristic of Zygomycota is that it forms sexual spores when hyphae fuse. And that sexual spore is commonly called a zygote. Now, a zygote is a very general term that means that it's the result of sexual reproduction in which each parent contributes half the DNA. So a zygote occurs in humans, it occurs in virtually all mammals, it occurs in many other animal sides, but it's also true that if you have a union of two hyphal forms and there's an equal amount of DNA contributed, then this is also a zygote. And so this, what you start out with is the spores that form these sporangium, and again you have a negative and a positive mating strain. You have one group up here that's the negative mating strain, another group that's a positive mating strain, and these come from the spores. And then the positive and negative mate, and there's an equal contribution of DNA until you ultimately form the next sporangium, which produces more spores, and the life cycle continues. Now this picture is a very interesting fungus called the, and I'm not going to say that word, but this is characterized by the fact that these spores form flagella. So you have basidiomycoses where the spores are produced in a club-like structure. You have the ascomycota, which is the spores are produced in a sac or held in a sac-like structure. You have the zygomycota, which is the fusion of two hyphae result in a zygote. And you have this mycota, in which the spores that are formed have flagella, and you can see these flagella right here. And the single cells here are also called chytrids, and they generally inhabit muddy water, and they are frequently saprophytic but also at times parasitic. The last phylum I'll discuss before we take another break is Deuteromycota. And Deuteromycota, you know what Deuteronomy is. Deuteronomy means second book. Well, that means Deuteromycota, for some reason they called this second fungus. And the Deuteromycota is interesting because it's a fungus in which they have never been able to identify the sexual reproduction that occurs in this fungus. Now, for this reason, Deuteromycota frequently has been put in a different category than the rest of the fungi. And because they are not known, they're also called imperfect fungi. So, one of the nice things about Deuteromycota is it produces a chemical that can kill other or inhibit other living organisms, and this is what's known as an antibiotic. Now, because the sexual reproduction is not known, Deuteromycota frequently are not, it's not known for sure whether they're in the proper phylum or not, or they should be considered a separate phylum on their own. Be that as it may, for this class, we will consider it part of the fungi. I'm going to stop at this point. We'll come back for Myxomycota in the third part of this lecture.